Hello everybody, welcome to YouTube channel Understood Biology. We're doing reproduction in plants and we have done the structure of inducium and the gynoecium, inducium in the male part and gynoecium in the female part. Now how fertilization occurs and the seeds and fruits are formed. So now we'll deal with this. For in the male part of the plant, that is in the inducium, the stamens are present, and stamens, each stamen have filament and anther. Anther bears many of the pollen grains, millions of pollen grains, and these pollen grains, they are to be shed on the stigma of the female flower so that fertilization occur. Pollen grain, they are the actually gametophyte. So each pollen grain, when they are shed, they are two-celled stage. In most of the plants, vegetative cell and the generative cell. However, in other plants, they may be shed at three-cell stage. Generative cell divides into two mitotically, and the haploid, they are haploid generative cells and these are vegetative cells. Then generative cell divides into two. Two male gametes are formed, and pollen grain is accordingly called as the male gametophyte. So how these pollen grains, they are carried to the stigma of the female flower, say stigma of the pistil. So how does it occur? The transference of pollen grains to the stigma of the female flower is called as the pollination. Depending upon its source, pollination is of three types: autogamy, geitonogamy, and xenogamy. Autogamy, when the pollen grains of the same flower, of the one flower, they are transferred to the stigma of the pistil of the same flower. That is called as the autogamy. There is no, it is pure breeding. There is no genetic difference in genetic combination. So that is called as autogamy. Then it is also called as the self-pollination. It is also called as self-pollination. And then when the pollen grain of the flower, one flower, they are transferred to the pollen grain of the other flower bearing on the same plant. That is called as geitonogamy. Otherwise, it is cross-pollination, because the pollen grains, they are transferred from one flower to the another flower, but the genetic composition of the plant is the same. They are transferred to the flower of the same plant. So they may be called as self-pollination also, autogamy also. So then is xenogamy. Xenogamy, the pollen grain of one flower, they are transferred to the stigma of the flower of another plant. That is called as xenogamy, and in the true sense, cross-pollination. So geitonogamy and xenogamy, you can say it is cross-pollination, and autogamy to say self-pollination. So how does it occur? In autogamy, it is there is necessity that stamen, male part and the female part, they should come in contact with each other to implement the pollination. What happens? Most of the flower, they are chasmogamous. What is chasmogamous? Open flower, which is visible, or four parts are visible. Open flower, they are called as. They are not in bud condition, then they are called as chasmogamous flower. In some cases, just as in case of Viola, Commelina, and Oxalis, two types of flowers there, chasmogamous as well as clistogamous. Clistogamous flowers, they never open. They are always in the bud condition. So stamens and the pistil, they are in contact with each other. So there is no chance of cross-pollination in the clistogamous flower, because they never open, and the pollen grain of the same flower, they are transferred to the pollen grain pistil of the same flower. So that is autogamous. So clistogamous flower, they are always autogamous. Then is how cross-pollination occurs. For cross-pollination, agents are required for cross-pollination. The agents may be abiotic or biotic. Abiotic means non-living agents, that is water and wind. Biotic means living agent, insects, bats, birds, and you can say these are the bats, these are the pollinating agents. So when the pollination is by air, the pollen grains should be to say produced in large quantity. They should be light in weight, they should be non-sticky so that they can be carried by the wind. And they are produced in large quantities because large numbers, because many of the pollen grains they are wasted in the way. So in such type of pollination, when they are carried by the wind to the from the stamen of one plant, male plant, to the female plant, female flower, it is called as the anemophilous. Pollination by wind or air is called as anemophilous. Anemophilous flower, anemophilous plant or the anemophilous flower, they may not be showy. They may not produce nectar even. They are small because they don't need to attract the biotic agents, that is insects, bats and birds, etc. So in wind pollinated flowers, they are small and their pollen grains are non-sticky, light in weight. They are produced in millions of numbers, and the flowers they bear the flowers are not showy. They don't produce any center nectar as in case of grasses, etc. So then is another abiotic agent is water. So near about 30 species of plants, they are water pollinated and they are mostly monocots. So the plants which are aquatic plants, which are grown in water, naturally there will be pollination by water, as in case of Vallisneria, Hydrilla, and in case of Jostera, seagrasses, there is water pollination. In Vallisneria, the flower, female flower, they grow up to the length of surface of the water. They come to the surface, and style is long. They come to the surface, and the male flower they produce pollen grains which are carried by the current of water to the female flower, and pollination occurs. In case of Justera seed pollination may occur under the water. So both of the flowers they are underwater. In case of some aquatic plant, just like water hyacinth and water lilies, they are aquatic, but the pollination is either by insects or by air, because they grow, their flowers they grow above the surface of water. This is all about water pollination. And then we come to the pollination by biotic agents. Insects, they are the most prevalent agents for pollination. Many varieties of insects, many species of insects, they help in pollinating the plants, flowers, that is just like bees, beetles, wasps, and moths. All these they help in pollinating the plant. So when insects they visit the flower, they visit the flower for their own purpose. They are attracted by the colors of the flower. They visit the flower for seeking nectar, which is their food. So the insect pollinated flower should be big somewhat, and should be colorful. Their petals are colored, and they should be producing nectar and any other substance to attract the insects. So there is to say flower provides some factors to them to attract them, and that's why there is mutual. They visit the flower for their own food, but they help in pollinating the flowers. So insect pollinated flower, naturally they are to say colorful and producing nectars, and some flowers which are pollinated by moths, etc., they produce bad odors, and as per their choice. Then some the pollination by insect is called as entomophilous. There are some plants which are pollinated by bats, that is called as a chiropterophilous. Some plants, they are pollinated by birds even, that is called as the ornithophilus. Then some flowers, in some flowers, they are site of egg laying for some insects. Such flowers, they are very, to say, large, just like Amorphophilus. The agent lays eggs there, insects, it pollinates the flower. And this amorphous flower is itself is 6 feet long. So this is all about insect biotic pollination by biotic agents. Then, pollen grains, I told you, they are very hard structures, and their outer covering is very hard, exine, and inner covering is entine. Outer covering is formed of sporopollenin. It is very hard, hardest substance of the word. It is not affected by, to say, any enzyme or climatic condition, high temperature and heat, etc., humidity. They are not. So these pollen grain, they have been found. Their fossils have also been found. In some cases, 10,000-year-old fossil of pollen grain have been found, and in other cases, 2,000 year old fossil of pollen grain is found, and this is due to the sporopollenin. Otherwise, pollen grain, what is their viability? Viability of pollen grain may be few minutes, that is 30 minutes, and as in case of grasses, it may be for months and years, for months as in case of leguminaceae, rosaceae, and all these solanaceae. So they say, then when pollen grains, they are shed, I told you, they may be shed at 2-cell stage, that is generative cell and the vegetative cell. The exine of the pollen grain is very hard, and intine is formed of pectin and cellulose. It is continuous layer. It produces the pollen tube. In the pollen tube, to say, the vegetative cells and generative cells, they are transferred. Generative cell is at the tip. So pollen grain is shed at this stage. So then when pollen grain is shed, when it is taken by any agent to the female flower, then what happens? The female part of the plant, the pollen tube enters, pollen grain enters through the pollen tube from stigma to style, then ovary, then to the ovule. In the ovule is present nucellus, and nucellus, nucellus has inside embryo sac, which is, you can say, female gametophyte. It has megaspore nucleus, that is haploid. This nucleus divides. This is the female gametophyte. Embryo sac is the female gametophyte. It is haploid because megaspore mother cell, it divides meiotically into 4 megaspore, and 3 megaspore, they are non-functional, they degenerate. Only 1 megaspore, it develops into embryo sac. It is present in nucellus. And then this megaspore nucleus then divides mitotically thrice. First 1 into 2, then second event 2 into 4, third event 4 into 8. But no cell wall are formed. 3 nuclei, they are present at the micropellar end, and 3 nuclei, they are present at the chaliceal end, 2 nuclei in the center of the embryo sac. So what happens is this, after their division, cell wall is also formed. Then 3 cells at the micropellar end are formed, 3 cells at the chaliceal end are formed, and 2 nuclei, they are not surrounded by a cell wall. They are present freely in the center, and this cell is called as the central cell. Polar nuclei, they are present freely too. All these, they are haploid. Alright. Then the pollen tube enters through synergid into the egg. The synergids and egg cell, they constitute egg apparatus. These synergids, they bear on their outer side a thickening, which is called as the filiform apparatus. This filiform apparatus It acts as the guide to the pollen tube, how to enter inside. So through the synergies, they enter through the egg cell, and one gamete, here the pollen tube divides, the generator cell divides into two. Two gametes are formed. One gamete will fuse with the egg and zygote will form. Another gamete will fuse with two polar nuclei. Then this primary endosperm nucleus is formed. So this primary endosperm nucleus, it is triploid and zygote is haploid. When there is fusion of egg with the male gamete, is called as syngamy. When there is after that fusion of two polar nuclei with another male gamete, primary endosperm nucleus, the process is called as the double fertilization. It is called as double fertilization. These synergids and antipodal cells, they are nourishing and they will degenerate afterward. So fertilization occurs. This is the ultimate product of the fertilization is zygote and primary endosperm cell. So further development into embryo is called as embryogeny. From the zygote embryo is formed. Primary endosperm cell, it helps in nourishing the developing zygote. So primary endosperm cell, it sometimes before the formation of embryo from the zygote, before the start of division in the zygote, the primary endosperm nucleus is triploid, I told you, and zygote is diploid. It starts dividing and redividing, and multicellular structure is formed that is called the endosperm, and this endosperm nourishes the developing zygote. Zygote divides and redivides by mitosis, and heart-shaped structure is formed which is called as the embryo. Each embryo, in case of dicotyledons, it has two cotyledons. To our cotyledon is attached embryonal axis. This embryonal axis, upper side of embryonal axis is called as epicotyle, and lower side of the embryonal axis is called as hypocotyle. The tip of epicotyle is plumule, which is the future shoot. The tip of hypocotyle is radical. It is the future root. And in most of the plants, by this embryo, it is further nothing. It is the seed. So what happens? Most of the seed, this end of embryo formation, primary endosperm is totally consumed, and the seed which is then formed, that is called as the non-endospermic seed or the X-albuminous seed, and as in case of pea and groundnut. However, in other cases, some portion of the endosperm is left, and such seeds, they are called as the endospermic seeds or albuminous seed. Non-endospermic X-albuminous or endospermic albuminous seed. So in some case, in case of this is the structure of dicot seed. So this ovule, the wall of the ovule, integuments of the ovule, outer integument, inner integument, they form the course of the seed course. The outer integument, it forms the, you can say, testa, and the inner integument will form the tegument of the seed. The cotyledons, they retain the food material. They are a source of food for the developing embryo. So at this stage, what happens? The seeds, they lose water. They get dehydrated. Their metabolic activity ceases, but they are alive. And then seed, they come into dormant stage. So when the seed come into dormant state, they can be stored for a long period. So they help in agriculture as well. So seeds can be used year by year. So because of their dormancy, their character of dormancy. And you know, in the seed, if you see the seed of gram, Bengal gram, or you can say, there is some pore at the beginning pointed on, that is called a micropyle. And a structure like here is present, hilum, from which it was attached to the funicle. Funicle was attached, And then in monocot, single cotyledon is formed. Endosperm is retained, as in grasses, in wheat, maize, endosperm is retained. And below the endosperm is alluron layer, and below the endosperm alluron layer is small cotyledon, which is called as scutellum. To the lateral side of scutellum is attached embryo axis, and similarly as in case of dicot, the above the cotyledon embryo axis is called as epicotyle. Below the cotyledon, it is called a portion below the cotyledon is hypocotyle. Upper tip of the epicotyle is the plumule, covered by a sheath of cells, that is called as the coleoptile, and similarly radicle is covered by outer sheath of cells, that is called as the coleorrhiza. So you can see this is all about the structure of seed. And after fertilization, zygote is converted into seed. Endosperm is consumed. When it is retained, then the seed are endospermic. In some cases, as in case of coconut, the endosperm, when primary endosperm nucleus divides, there is few nuclear division. No cell wall is formed, only nucleus divides to so many nuclei. And after some time, the walls are laid down to some of the nuclei, and cellular endosperm is formed. You know, when we take the coconut water, that is nothing, it is the nuclear endosperm. It is a liquid endosperm, and it's a very nourishing, and the white edible portion is the cellular endosperm. In some seeds, this nucellus also is retained. Some part of nucellus is retained. They are formed the perisperm. Above the seed coat is the perisperm, and the seeds are called as the perispermic seed. So this is all about fertilization. And you know, ovules, they develop into seeds. You know, the walls of the ovule, they will form the testa and the tegmen, and zygote will form the embryo, etc. Ovary will develop into fruit. Ovary will develop into fruit. The wall of the ovary form the pericarp. That is the covering of the fruit. Pericarp is further differentiated into 3 layers, in some cases into 2 layers. Both layers, outermost layer is epicarp, middle one is mesocarp, and innermost is the endocarp. Endocarp is mostly stony in most of the fruits. Just you can say in case of mango, the outer peel we peel out, that is the epicarp. The fleshy portion, yellow portion we eat, that is the mesocarp. And the inner seed covering, the hard layer covering the seed, that is called as the endocarp. The seed is formed from, fruit is formed from the ovary. But in some cases, along with ovary, thalamus of the flower is also converted into food. The fruit is developed from thalamus along with ovary. Such fruits, they are called as the false fruit, as in case of apple, strawberry. So the fruits which are developed from the entirely exclusively from the ovary, they are called as the true fruits. And the fruits which are developed from to ovary along with other parts of the flower, just like in case of apple, strawberry, thalamus is also take part in the formation of fruit, then it is called as the false fruit. So this is all about fruit formation. Then you can say there is what is apomixis and what is parthenogenesis. In case of some plants, what happens? The cell of one of the cell of the nucellus, which is diploid, it acts like embryo, and it develops into the embryo. So there is no fertilization. The cell is already diploid. It will develop into the embryo, and there is no fertilization. Such type of seed formation is called as the apomixis. When the seeds are formed without fertilization, it is called as the apomixis. And some cases, what happens? More than one embryo is formed. As in case of citrus, many of the cells of the, you can say, nucellus, they start like embryo and divide and divide to form the seeds. Many seeds are formed. So such process is called as the polyembryony. Many embryos are formed inside the nucellus. So parthenocarpic fruit or parthenogenesis when there is no fertilization, but there is development of seeds, there is development of fruit. Ovary start developing into fruit. Ovary matures into fruit. Ovary ripens into fruit. The process is called as the parthenogenesis, and the fruits are called as parthenocarpic fruit. So this is all about to say fertilization and post-fertilization stages. Then is seeds are formed. Seeds are required to be dispersed. Seeds are stored by the agriculturists, and some wild plants and some other plants also. Seeds are dispersed by many methods, by animals, by air. Just in case of calotropis, seeds are very light. Their winged seeds are produced and they are dispersed by air. And skull, it takes the fruit of the plant and then takes some of the part and it goes away and split all these broken fruit here and there, and it also help in dispersal of fruits. Birds, they take the fruits and some through their fishes seeds which are not to say digested, they come out and they are also help in dispersal of seeds. So what is hybridization? Outbreeding experiment. What are outbreeding? To say, the pollen grain of one flower, they are transferred to the pollen grain to the same flower, which is called as the autogamy. If continuous process of autogamy is there, self-pollination there is no improvement in the variety. There is no chance of genetic changes. So the ultimate products, that is seed and fruit, they may be weak, and we don't get the desired characters. So in pollination, cross-pollination, however, there might be chances of great chances of to say mixing of characters. So when what happens sometimes these the seeds, they are produced, hybrid seeds are produced and hybrid plants are produced to combine the desired characters of the two plants. What happens? Pollen grain of one plant, pollen grain of one flower of one plant with desired character, it transferred to the pollen grain of the other plant with desired character, and then hybrid seeds are formed which are used for further production of the plant with desired characters. So how it is done? What happens when we want to transfer the pollen grain of one plant to the transfer to the pollen grain stigma of the flower of the other plant? The what is done? Pollen grain of that receptive plant, they are removed. They are removed, and that process called as emasculation. Then the pollen grains of the desired plant, they are transferred to the stigma of that plant because they have been removed, and then they are repacked. They are bagged from outside so that they may not be contaminated. So this process is called as the bagging. So then is pollen pistil interaction. What is that pollen pistil interaction? Pollen grain of one flower, they are transferred to the pollen grain of other flower of other plant. They may be transferred to the same flower. So there should be compatibility between the pollen grain and the pistil. The stigma is receptive only to those pollen grain. They allow the germination of pollen tube, formation of pollen, only those pollen grains which are compatible to. If the pollen grains are not compatible, the stigma won't receive it. It won't allow it to germinate the pollen tube. So that is called the compatibility. And if the pollen grains of this compatibility, it is itself is in nature. In some cases, the pollen grain of the same flower, they are not compatible to the pollen grain of the pistil of the same flower. What happens in that case? To avoid the pollination, either the pollen grain anther will mature earlier, dehisces earlier, and stigma will mature late. Either stigma will mature earlier, the ovary will mature earlier, and the distance of anther is late. So pollination, autogamy is avoided, and cross pollination is there. So other is their position in the flower is different. So anther is on this side, stigma is in this side. They can't come in contact with each other. Again, then self pollination is avoided. This is called as the incompatibility. And when the pollen pistil of one plant of one flower of any plant, it receives the pollen grain of the plant of the other flower, then if it is compatible, it will grow, other it will not. The growth of the pollen tube, then movement of the pollen tube inside the ovary, ovule, then through the synovium into the egg, and fertilization and double fertilization, that is called as the pollen pistil interaction. It will only happen in between the pollen grain compatibility, if there is compatibility in between the pollen grain and the pistil. This is all about pollen grain and to say pistil interaction. Then hybrid seeds are formed, but it's very difficult. We get the desired character, but it's difficult to maintain the characters in the next generation. Every year, every time new hybrid seeds are to be formed, because during their growing in the next season, there will be segregation of the characters, and characters will be lost. So how it's possible? The most prevalent method is by apomictic. Apomictic seeds are those which are formed without fertilization. The plant is having, when the hybrid seeds are formed, then in the next generation, the apomictic seeds, there is no fertilization and plants are raised. This is all about reproduction plan. I think you might have followed it. Listen it, like it, don't forget to like it, and subscribe it and share it with your friend. Okay, have a good day. Bye.